Hi, everyone. I'm Salma Quraishi. Welcome to Neuroscientists Talk Shop, the University of Texas at San Antonio's neuroscience research podcast. Today is March 25th, 2021, and we're talking with Kay Tai, who is professor and Wylie Vale chair at the Salk Institute and investigator in the Kavli Institute for Brain and Mind at UC San Diego. Hello, Kay.
Hi.
So the TIE lab broadly examines the neural circuit mechanisms of motivated behavior, valence, emotional states, and more recently social interaction. Her group has a big, ever-innovating toolkit that includes cellular resolution recording and imaging, behavioral assays, projection-specific optogenetic manipulations, and computational tracking and analysis tools. And today in the Zoom, we've got a couple of our own motivated behavior specialists. We've got Matt Wanat. Hi, Matt.
Howdy.
And we've got TIE Lab alum, Tony Burgos-Robles.
Hello.
Hey, Tony. And of course, we've got Charlie Wilson, as always. Hi, Charlie. Kate, your recent work has really boldly taken the circuit mapping framework mastered by your lab in individual brains and pulled the lens out onto brains that are interacting with one another. And you've managed to do this without drifting too far from cellular and biological level resolution and questions. So talk to us about treading this intersection between thinking about the brain from a cellular space to an ethological space. Is it as simple as saying that social interaction is just another dimension of the environment that we're always trying to manipulate as experimentalists, or is it something broader and deeper in terms of the questions that you're after?
So such a great question. And as you were asking the question, I thought of some, I started to think about the way the field works. And it's like, you've got these different subfields, and neuroscience is such a fun landscape because, you've got like reduction is sort of synaptic physiology. Wanat and I used to be in a synaptic physiology lab together. It was just like 12 rigs, everyone's doing one thing. And then, moving to circuit stuff, moving into systems and like more different kinds of frameworks and crossing those boundaries. I mean, I think you can think about like fingers coming out, like where it's like, okay, psychology, right? And then it really diverged from neuroscience. And it's almost like this big wall between them. And then there's all this space in the middle. You could be at the forefront in a lot of different ways. And you can be a technological leader. You could be at the bleeding edge of pushing some concept forward, or you could also be like connecting the dots and being that connective tissue in between these webs. I guess, I've never posed before, but your question just made me think like, oh yeah, what is like my style? And, I never really thought about it before, but I think it's making these types of connections are just something that's what my brain does. And that's like, oh, like, that's how I think about things. And then, that's just kind of what naturally comes out. So, I don't know if that was the answer that you were looking for.
I'm not looking for any, I'm looking for your truth. So what's interesting is that your work with Jillian Matthews, it seems it's on social connectedness and isolation. And I guess now also with Lee, sorry, I don't know his first name.
Christopher Lee.
Christopher Lee, right. It seems timed so perfectly right now for our period of global isolation and concern with interactions. So it's really interesting to look at the way you've taken, so it's essentially a study on dorsal raphe dopamine neurons. And it poses this nucleus within this space that creates kind of this homeostatic mechanism for social interaction. So this framework kind of says animals have an individual defined set point for social interaction, and deviation from that set point is detected and triggers an effector to correct course. There's so many cool things that I can ask you just based on that part alone, but could you just start by saying something about the dorsal raphe dopamine neurons as the effector? You pose them as the effector mechanism in this, and then I really want to talk about what defines set point in your mind and how you
that's great summary of my talk. I think that the dorsal raphe story is just such an interesting twist and turn of how we got there. I mean, I never thought I was going to be studying social isolation. It wasn't an idea that I had. It was just following the data. And, it really came about serendipitously. Actually, So Wanat would know. So Wanat and I were both in the Banshee lab at the same time. And, the big superstar paper that had come out, Mark Unglis is, I guess, is like our neuroscience brother, Wanat, right?
Yeah.
Like older brother we didn't overlap with. And he had this huge paper about cocaine and dopamine neurons where, you give animals one injection of cocaine and then you get LTP. And that was, like this big Nature paper and super influential in the field. And Jillian worked with Mark and did this cocaine experiment in the dorsal raphe nucleus, a brain region that's mostly known for serotonin neurons, but also has some dopamine neurons. And at first it was just intended to be like an incremental study of like our DRN dopamine neurons, like VTA dopamine neurons or not. Like that's kind of, do they also respond the same way to cocaine? And then the saline group was different. And we realized it was like all these other manipulations that had gone into it. It wasn't, well, Jillian realized that, it was after you inject the animals with either cocaine or saline, you move them to the cage by themselves so they don't like freak out the other animals. I don't really know. That's just the protocol. We're just doing it. And then, she had she had that control instead of naive animals. It was a really well-designed experiment. And she trusted it and followed it and found that these dorsal raphe nucleus dopamine neurons undergo plasticity when animals are socially isolated. It's nothing to do with cocaine. It's nothing to do with novelty. It's the isolation. And they become more responsive to social stimuli when that occurs. And when you activate them, they mimic a loneliness-like state in that animals become more motivated to seek social contact, but it's an unpleasant, aversive state they want to escape from. And it's something that we see this correlation with social rank. And so that got us to think about social rank. And, we don't know for sure that it's an effector system. It could be the control center, but it sort of reflects already the recognition of a deficit. But, when we think about a control center, I don't know. Yeah. I don't know if I would expect that to be localized together in one nucleus or distributed across a macro circuit. We don't know yet.
So,
go ahead.
Okay, I was curious in Jillian's work and a lot of the work that you were talking about today, have you been looking at just males only or male and females? And you probably can see where I'm going on this because Tom Cash had a paper that just came out and was talking about some intriguing sex differences that they found when they were perturbing both optogenetic and chemogenetic activation, the dorsal raphe ventral PAG dopamine neurons. And I was curious, do you think that the same rules of, more generally sort of big picture, is isolation going to be affecting males and females in rodents the same way? Do we know that? And also sort of social structures when you end up having both males and females together. I know it's big question, lots of directions to go on that, but I guess, have you started to dabble in the sort of sex differences, or are there any sex differences?
Great question. So within our lab, we with Jillian's paper, we did not do sex differences. That I don't have a good reason for that. That just was the time of where we were at the point. And then we started thinking more about social rank stuff. And so I think in our hands, at this point we are able to get ranks in females, but when we were at MIT, we were unable, it was very unreliable if we would get linear ranks. So for the rank project, we didn't do females because I think female hierarchies are fundamentally different. Like the behaviors tend to be more, there's a little bit more turn taking rather than like something more despotic where there's an alpha that really dominates, the dynamics are different. And maybe that, there could be a lot of other factors that contribute to that as well. This paper is great. And I love, Tom Cash's work and thinking about, it is a different subset of DRN dopamine neurons that like mediate the social component. We have now identified that is dorsal raphe nucleus to central amygdala. Surprise, I don't know. And not BNST or the basolateral amygdala. We haven't looked at ventral. We haven't looked at I don't know what specific subsets Tom has investigated totally, but there is some overlap overall in the non-specific DRN dopamine neurons that for nociception. So like, physical pain versus social emotional pain, I guess. The idea that there could be a convergence is just so cool because I guess I don't know that I would have intuitively assumed that. Yes, there's a linguistic link, but aside from that, it was not obvious to me. And it was sort of interesting and, that there might be some convergence of those signals, those different strongly. So, if you think about it more as a teaching signal, maybe that makes more sense, but that's neither here nor there. For sex differences within dorsal raphe nucleus, there our collaborators in Korea have found that there are sex differences, but that's still unpublished, so I won't get into the details of it. But yes, definitely there is.
I guess I have a follow-up questions related to this. Like thinking about Matt's question on individual differences, basically, right? So, at least, in the case of humans, we undergo many stress situations across our lives, right? Different individuals get, some detrimental effects of those stress experiences, but many other individuals develop coping mechanisms, right? That makes them more resilient over and over as they keep experiencing even more stress. I'm just wondering, also in light of the pandemic, in which basically all of us were forced to socially isolate, right?
Mm-hmm.
Where do you think like we can start studying these kind of differences? Like resilient individuals versus non-resilient, and what do you think about that?
Yeah. So there's so many different things. There's so many different things in this question.
Sorry about that.
No. I mean, I think that, okay. So in some ways, the pandemic, I think in many ways for this line of research, the pandemic was great because it made it a priority. Nobody wanted to publish social isolation papers at first. Like it was like so difficult that, Jillian's PhD paper got rolled into her postdoc with me because it was just, we couldn't publish it and no one cared. And then all of a sudden everybody really cares. So that's great. I'm glad that people care because it's important. But I think that the first of all, the type of isolation that is experienced is very different. Like there's this concept of social distancing, which is in itself a different type of experience. And so I think we've got to we have to categorize them. And I think one important division is isolation that you choose versus isolation that you feel is you're excluded from. So that's something that's sort of been known for a while. But what about when there's like a third? Then it's not like you're not left out of this party. You know what I mean? It's not like there just are no parties. So, there's a feeling if you weren't invited to a party, that feels bad. If you were invited to a party, but you chose not to go, it doesn't feel as bad. And then like, if there just was no party, what is that? So it's kind of a completely different situation. And there have been a lot of cognitive, human imaging studies of different types of exclusion and what that means, like, oh, there's a ball game and they don't pass this particular subject, and then how do you experience that exclusion? And I think this is going to lead to a lot of different variations of levels of social distancing or social isolation or deprivation of social contact. The other factor is that in terms of the timelines that we have previously thought were ethical or non-ethical, because I mean, after the Harry Harlow experiments, with the social isolation of young monkeys, and it was after that, there was that was like this beginning of PETA and like, all of this ethical welfare of animals, and that that just has this stigma of being so like confinement is torture, et cetera, et cetera. And I think having an experience like this might make us rethink the relative importance versus the stigma of something being harmful. So, I think before there wasn't as much of a reason, what would be the benefit of studying this? Nobody understood the benefit. And so it just, nobody could do social isolation experiments, certainly not in primates, certainly not in humans, of course. But now it changes it.
Yeah, totally. Myself, like as we spoke this morning, like we began a project on the effects of social isolation and even how learning processes and detecting threats and detecting safety options in the environment are changed just due to some social isolation exposure, right? So I guess also my question, and I think, like within your answer, you mentioned something about, like there's different kinds of social isolation, either if we choose or not. I was also thinking about when I formulated my question about like patients, for example, that got COVID infections, right? Like
They were
those,
actually isolated.
yeah,
Yeah.
they are actually isolated, and it's so important that we understand, like the neural basis or how, just this isolation experience affects very dramatically our brain function, in general.
I was actually curious, have you guys played, I mean, Kay, when you were sort of describing the sort of different gradations of isolation, I mean, it's likely what we have, or in the laboratory setting, you've got there's a party going on and you're not involved. Like, have you ever sort of taken the isolated animals and made sure they're in their own segregated room with only other isolated animals, or are they sort of in the housing situation where they can do that? Because
And
it
hear
really
all the
just,
parties that are happening without them.
yeah, I mean, actually, I mean,
Oh, that's such an interesting idea. I love that idea.
anyway, it's just something.
Man, oh, how do we do it? Like, get another room.
Yeah,
We have to
it's not
build a
going to be
little
cheap.
cubicle. I mean,
Yeah.
basically, no, that's a really important. I mean, because you're absolutely right. Like those ultrasonic vocalizations, those social calls, absolutely, all the other animals can hear it. You know what I mean? I'm sure that they can. You definitely can hear everybody else having their conversations while you're in your cell by yourself. It's like, yeah, that's a really interesting question. The proposed experience that we were thinking about in marmosets would, because you can't isolate marmosets, like there's no way that that will be allowable. But you could distance them so they're still like visually and like auditorily in contact, but at a distance. I think that type of just like remove the physical component and then keep everything else is maybe something useful. It's not quite like Zoom, but I mean, Zoom is pretty good audio visual. It's not perfect. It's not the same as being, separated, but it might be more analogous. I don't know. I think moving one modality at a time might be useful in terms of understanding what's going on.
Can we talk about rank a bit? Because embedded in
Sure.
all of that, you mentioned rank, and rank is what determines whether the valence of isolation is going to be positive or negative. So if you're low on the hierarchy, or this is like an intuition that you have to think about whether the valence is going to be positive or negative, right? So
actually, for the positive or negative, we've been only thinking about it in terms of acute or chronic isolation. But you're right that like rank and isolation, it's interesting to think about the interaction. So talk about where rank sort of comes into the dorsal raphe studies, because that's some work that you've done. And then there's a lot of work on medial prefrontal cortex encoding brain states that are associated with rank. And I really hope that you guys could talk a little bit about that, because that's work that is so interesting and I don't know anything about it. And, because rank itself is so context specific, right? In my house, I'm the king. Here, I'm not. So are these global, like how do we imagine that these are sort of globally entrenched things, and how sort of flexible are they? You can change rank within a social context. And what does that mean? Humans change ranks many times throughout the day, meeting to meeting, lab meeting to like faculty meeting. It's like boom, 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 constantly changing. But great question. So it's, I guess, okay, the wild speculation thing that I had was that, we're trying to think about DRN dopamine neurons representing a loneliness-like state, but loneliness is subjective. So like, how could we even get at this? So we just started trying to look at social rank because it was a way to access subjectivity. And we did see this super strong correlation between the optogenetic manipulations, all the manipulations we did, and basically all the effects are being driven by the dominants. They're all strongly correlated with social rank. Subordinates stimulating these neurons doesn't really do anything. And the subordinate neurons, maybe had a little bit, this is evidence is kind of shaky. One look at this, we found a correlation, and another look wasn't replicated. So we'll see. But initially we saw maybe that there's more TH in dominant animals than subordinates. And so like, okay, maybe they're not having it. Like, why would stimulating the same neurons in one population of animals be, why would that be different? Like, why would it only affect dominants and not subordinates, and like social context tasks and even place avoidance tasks? It's like, that is nothing that's social necessarily. So we try to think about what could be going on there. So then we started to think about social rank and how, you can't really think about the homeostat without understanding how social information is detected. And we can't think about how social information is detected if we don't know the source, because the source of the social information really matters in how you would interpret it. So understanding rank became like, oh, we really need to do this first.
So just a technical question. In those experiments, was dominance and subordinate status worked out within that context in that experiment, like whoever won the battle, or like, are these sort of larger structures that you're categorizing?
No, we ranked them. We did 2 ranks, and everybody had a rank in their cage, and then we just correlated like the degree, the delta of change with light on versus light off, like how much light changed behavior and social rank, and it was very strongly correlated for every task, like place avoidance, channel rhodopsin, gain of function, social and muscle function. So that was like, rank is doing something. And rank seems like, it matters, because if you have a social need state with that female or whatever, I know that I'm not, like, I'm gonna get beat up by this alpha if I try to mate with this female, then maybe I would suppress that. Maybe I don't want that drive to be so strong.
So, okay, I was wondering if maybe you could talk a little bit about how you've sort of been moving into the new direction of being able to sort of record activity from multiple animals at the same time, which I think is a really sort of exciting way where, a lot of times, we're focused on one animal in a contrived, circumstance, but it really is moving the field forward, by leaps and bounds, by, having multiple animals being able to interact, or at least 2. And, what you've been doing and what you've been finding, I guess, with that, and sort of devising those experiments, which are, quite elegant as far as being able to look at the interaction between one that, might be more dominant versus another one that's subordinate.
Yeah, no, those we tried a bunch of different things. I mean, honestly, it was like, I was kind of thinking maybe imaging would be ready first, like wireless imaging would be ready first. But then talking to Matthias Carlson, who Wanat and I used to go climbing with back in like grad school 20 years ago, or I don't know how long it was, but long time ago. And his company makes these wireless, head stages for in vivo ephys, and there's just a little battery. So that's what we've been using, and it works great. And it allowed us, we can do many more animals. I think it was all just about, like, now that the computer vision tool finally works, I would be comfortable using a task with more animals. But at the time, we weren't, everything was kind of being developed in parallel. It was like the hardware, the software, the task is like all just, hope all these things work out, because the project won't be possible otherwise. So that's kind of how we got to it. Is absolutely, we should be doing it with way more.
Yeah,
I think, oops, sorry.
Yeah, that's totally, yeah, I was hoping you would get to, thank you, Wanat, because this idea of just of social interaction being this environmental thing that we're manipulating is really not the question, right? It's looking at brains interacting and with one another and seeing the changes, relative changes.
No, so towards your previous question, Salma, thinking about how your rank changes rapidly. So we've been doing this is like a sort of a follow-up to this project where we record the animal with one, let's say we take an intermediate, we pair them with a dominant first, then they're by themselves a little bit, then they do another session with a subordinate, and then the next day we do like, control for order. What we can see is that, how does the representation shift from like, just minutes to minutes of what's going on there. We've also been looking at absolute rank in the cage, and whether it's all relative rank that really matters, or is it absolute rank that matters. To be PFCs representing relative rank, which makes sense. Like, it's not even clear that we necessarily have an absolute rank, if that makes any sense. It's like, what is my rank? It doesn't mean anything without the context of a social environment. At home, I'm an adult, I'm a parent, so I'm a boss. In my lab meeting, maybe I'm the alpha, but at like other faculty meetings or different conferences, like maybe I'm just, whatever. You just kind of have to snap between these different situations so rapidly. And I think it's possible that we don't. It doesn't seem like we store absolute rank in the prefrontal cortex. Maybe we store it somewhere else. I don't know. It's very preliminary. So we'll
So
see.
do you
if it all
imagine
holds up.
in the context of the tube test and where you have, or in the, the arena where the two animals are going at it and you have sort of multiple losses piling up. I don't know if this ever happens with a dominant and a subordinate.
Totally.
Could you see the context shifting, like the dominance shifting within, I don't think you've done this experiment, obviously, because I don't know if you have the prefrontal recordings of multiple losses in a row. Like, could you see?
We do. We see there's a lot of interesting patterns. There's often something where the dominant, it's kind of like, so if you look at if you break that decoding accuracy up into subordinates and dominants, usually people ask a question, so I just kind of save it for questions, but the dominant accuracy just like stays high the whole time. The subordinate accuracy, it's still like above chance, but it starts out kind of low and then like catches up to the dominant later. So my speculation is, and like also watching the videos, you can see that there's active wins where they're like both like pushing at the fort, and there's passive wins where one animal is just like in the corner grooming and it's like the other guy's turn. And so this is really interesting, but I think what happens is looking at the dominant basically to see their level of engagement, and then based on the level of engagement of the dominant, which is just like, I'm either trying to get this reward or I'm not. And then if I try to get it, I'll probably get it. And if I don't try to get it, I obviously won't get it. And that's like what the decoding accuracy comes from in the dominants. The subordinates, it's like, there's a little bit of that. There's, they could be just inattentive, but if they see the dominant not engaging, then they're like, ooh, this is my chance. And so you can see the decoding accuracy like really shoot up after the cue. Like if the cue goes on and the dominant's not going for it, they're like, woo, here's my chance. And then you can see them go. So it's interesting. I definitely think there is this asymmetry in the dynamic between the subordinate and the dominant.
I guess my next question is related to this because I was fascinated to see all this new analysis, computational analysis, to decode the activity of the medial prefrontal cortex to determine how the animals are performing during this reward competition task, right? And you mentioned, that you could go farther, like 30 seconds before, and the decoding accuracy is still as good as, like immediately prior to winning, right? So I'm wondering if, that's just a result of, the actual winning or, like the media history of what just happened in previous trials. And I think it nails to, goes back to Salma's question, or is it related to the overall activity in those dominant animals forever? Like I'm thinking if you have done any recordings even prior to training the animals during this competition task to see if, like the PFC activity can already predict, how are these animals going to perform, like later during the competition task, right?
That's an interesting idea. I mean, the way that we do the analysis, that wouldn't be possible because you would need training data from before. Right. Or maybe not. Maybe you could test it on, use current training data and then test it on old data. But then there's no outcomes. I think you need the behavioral outcomes to be able to test it. The way that we're doing it is we're decoding, we're like predicting trial by trial. So like maybe for rank, I don't know. We're also doing this rehousing experiment because, I alluded to the issue, which is a problem in the field that I'm guilty of this too at this point, where if we have neurons that respond to the president of the United States, for example, is that a rank neuron or is that an identity neuron? We don't know until you show me another president. You know what I mean? Like until that person isn't president anymore, or you show me like lots of presidents or something. We have to dissociate the rank from the identity. If neurons in my brain fired to Barack Obama when I met him when he was president, and then like a bunch of the neurons didn't respond after Trump was president or Biden's president, then like, that would be a rank neuron. But that experiment is non-trivial to do. So that's what we're doing right now. And the way that we're going about it is rehousing animals so that they will have different ranks and changing their ranks, essentially. Right. Finding ways to separate rank and identity. But
so can you, go ahead.
Well, I think following up a little bit on what Tony was saying there, like one of the most fascinating things is 30 seconds before the trial begins, you can look at the activity of what's happening in the prefrontal cortex, and you have great accuracy of predicting what the outcome is going to be. And you mentioned you couldn't go back any further. And I was curious because it was the preceding trial. And in some ways, I wonder, is it perhaps the subjective experience of what happened on the previous trial that's sort of driving, how good was that reward? Now, did I really like it? You know what, I'm going to fight for it now. And I guess it's sort of difficult questions, but I guess, can you even go back further and look at sort of, is the reward, the activity to the actual, preceding reward, is that then serving as the, not to say the gate, or at least it's the setting, it determines, where you're going to be. Are you likely going to fight for it,
Yeah.
or are you going to be chill out? And I guess that also potentially could pull out looking at trial by trials throughout this, because I would imagine, and I don't know without this, but like early on in a session, maybe a more dominant, maybe more aggressive. And then after maybe they get their fill, they'll be like chill out, and there might be more subordinate wins later on. And I guess, does that bear out? And sorry, that was a many layered question there.
No, I think, so for our experiments, we looked at that, but we didn't see anything really, really robust in terms of streaks of wins and losses. The way the task is, it ends up not really being significant. But the work from Hailan Hu really convincingly, I think, shows that there's a winner effect, or maybe it's more accurate to say a loser effect. That winning doesn't necessarily make you win more, but the losing does make you lose more, like make you give up more easily. So I think that's interesting to think about, and maybe a winning mindset's more fragile than a losing mindset. I don't know how you want to think about it. Like from a computational perspective, I think about it in terms of like attractor states. Like, is this attractor state gonna like be something that like holds more stably than a winning, more fragile sort of mindset or representation.
Although you would expect that an unexpected loss for someone who's on a winning streak would be especially disruptive, just intuitively, right?
Yes. I mean, I think it's not unrelated to like classical learning theory from what you would expect. But so, yeah, I think that is something that occurs. I don't know. I mean, our this particular task was, I think with the tube test, the winning is like maybe more salient. I was gonna say humiliating, and I was like, that is not very scientific. But it's like you have to walk backwards for a long time. Like you're losing for a long time. Whereas like if it's the reward competition task, it's just like, okay, you didn't get this one, but there'll be more. There's like so many more trials. Whereas tube tests, there's fewer trials. It's just a longer task. So I don't know. It's interesting to think about, but yeah.
So I wanted to get. I'm so glad you have a lot of energy, because this is going to be a long one. But I wanted to get to the difference. It seems like I don't know the literature, so maybe you can explain to us the difference between acute and chronic isolation. In the few things that I read seemed almost arbitrary, the way it's chalked out for rodents. But yet the outcomes when reintroduced into a social setting are so different that I guess it's a week versus more than a week is chronic versus acute. Maybe you could explain that. Talk about at a cellular synaptic level, the adaptations that you imagine could be responsible for this kind of thing, or how you're thinking about this adaptation, because you've called it an adaptation. And I don't know if you mean that in a literal synaptic sense of adaptation, or whether this is sort of behavioral.
I definitely think there is some sort of synaptic adaptation. We don't know what it is yet, but I mean, it's likely. There's an acute synaptic adaptation, and then sort of something that looks like a very homeostatic kind of time scale of maybe a little bit slow for homeostatic, but I imagine that is likely true. So we certainly have plasticity adaptations with on the scale of acute stuff, and acute and chronic, it's totally arbitrary in the field. And also, I mean, is it even time per se, or is it correction effort? And so, I think all of these things depend. And, given that different animals have different lifespans, chronic for a fly is not going to be the same as chronic for a human. And even a human mouse, like, human lives 80 years and a mouse lives 2 years. It's like really not, I don't know how to compare these timescales across different species of animals with such vastly different reproductive and lifespans. So I think maybe though within a species there is a time constant. You have a circadian rhythm. Maybe that is like how the allostatic load builds up, right? Where there's some amount of evolved set point for keeping track of time. I suppose that's possible. It seems kind of more likely to me that it's correction effort though. Like you're making calls or you're venturing out of your burrow. You're trying to, meet mates. It takes effort. And, if you have a lot of failures, you might decide that the cost benefit ratio just isn't worth it. And, with some updating, it isn't worth it anymore. And that's the possibility. I think in terms of the homeostatic set point adaptation, it's where would this form? What would trigger the adaptation? Would it be gradual? I mean, the behavioral change seems sort of step function like, you try every day until you just stop, right? Or whatever. Maybe not totally. It doesn't seem like linear. Whereas if it's an allostatic load kind of model where it's drip, drip, drip into a bucket until finally, you snap or whatever that means, or make your adaptation. I think that is also possible. We just haven't systematically worked this out. People have used isolation, not to necessarily explore the time course, but for chronic isolation as a model for schizophrenia, as, early life stress model, just as something that is sort of a sledgehammer, so to speak. And we haven't gone through to study the time scales of what is exactly changing at roughly a week. A lot of things do seem to change in our hands anecdotally. There is something around, the week-ish to 2-week mark that seems different, but there's genetic adaptations. David Anderson, Moriel Zelkowski from David Anderson's lab has shown that TAC2 may have a role. So there could be some genetic changes that occur, some epigenetic changes, on lots of possibilities, and it's probably all of these answers are contributing, most likely.
So there's obviously only a fixed number of hours in a day, but there's so many sort of variations on this theme of implementing sort of isolation that one can test. But I guess a couple that sort of popped out to my mind was, have you looked at sort of intermittent exposure of isolation, then back, isolation, back, and I guess sort of influence from the drug addiction literature of how intermittent access itself can end up having a more robust sort of phenotype, and a lot of models of drug addiction in some ways that could be sort of traumatic. And I guess the other thing I'm sort of thinking COVID of lockdown, you can go out of bed, lockdown, sort of those cycles. And I guess the other thing is juveniles as well, where, isolation particularly, I mean, from a translational perspective, isolation during adolescence can be, very traumatic. And, have you looked in, or anybody else for that matter, looked in how isolation is influencing behavior from sort of a developmental standpoint?
Great question. Byung-Kook Lim, my colleague at UCSD, has been looking at this and has looked at social isolation when it's predictable. I believe, versus when it's not. So the analogy he uses is like, if you're dropping your child off at daycare, that's like a very reliable time. There's gonna be, it's like very predictable. And if it's reliable every time, it's not a big deal. It's like if mommy's late, then there's like a huge stressor. Or if you don't know when you're gonna, with COVID, it was very different. It was very unpredictable. Everyone's like, what's gonna happen? When can we go back to work? What's happening? And then it was like different, just all over the place. So that was the uncertainty, I think, was very difficult to deal with, in contrast to something where like, here's the schedule, here's the plan, adjust. Whereas, you don't know if you should be adjusting. What should I be adapting to right now? Because it's sort of changing a lot. I think that makes it actually more difficult, for your brain to adapt, because it's not clear what strategy should be used. So I think anything predictable would be a better way to reduce the stress. That's very similar to drug addiction, right? Like it's the unpredictable intermittent access. If it was reliable, then it's okay. You have to have some days where there's lots more and some days there's none. That's what makes it work. It has to be that variability that makes you have this like hoarding instinct, so to speak. I don't know what the right word is, but
so how do you model that in this homeostasis flow? I mean, is that just is that another node in that, or how does that
yeah, no, that's really interesting. So we have not looked at that. We're not working in juveniles right now. We're mostly focusing on adults. I think so I don't know, the other answers to Wanat's question, but in terms of the interaction with many other components, one thing we are looking at relevant to what you were just bringing up is actually alcohol drinking. How does, alcohol sales were like through the roof during the pandemic. It was like hate crimes, screen usage, alcohol usage is like all like through the roof. And social isolation, first of all, there is a correlation between drinking and social rank. But across the board, all ranks of animals drink more when they're isolated. And so we're just kind of looking at that and looking at how prefrontal cortical neurons might change longitudinally across different phases of isolation that could predict their development of compulsive alcohol drinking behavior.
I guess I have a question about, like possible future directions and to understand better all these factors contributing, because you mentioned the predictability of the stressor, right? But also a huge factor that, has been investigated with other forms of stress is the controllability over the stressor, right? Which, just thinking about all this work from Steve Mayer in Boulder, right?
Yes.
That, even there are different subregions of the medial prefrontal cortex playing in very important roles in encoding this controllability over the stressor, right?
Yes.
So just wondering how, if any ideas come to mind about an experimental design in which animals are able to control a social isolation stressor. Maybe difficult, right? Maybe difficult to achieve, but
yeah, that's interesting.
Yeah, I guess it relates to the question we were talking about earlier about different types of social isolation, right? Is it forced? Are people, especially if they are sick, like there's no way they can exit the room or the ICU, you know what I mean? So
I think that one's so bad too, because one of the things that correlates with perceived loneliness or social isolation is increased severity in response to a viral immune challenge. So like, I mean, it's probably the worst thing that you could do for someone with COVID is to socially isolate them. I mean, I understand the reason of why people do that, but oh, that's rough. So
yeah.
You just keep throwing random questions. I was
we
going to
all
bring up
stated-
autism, but I'm not going to do that because that would be mean. But you have you done some work in prairie voles? I saw some stuff cited in the review, but I guess that was
no,
general.
that's work from other people's labs. I mean, beautiful work has been done in prairie voles, and talking to Zoe Donaldson about the difference between social isolation and loss, like of a pair bonded partner, for example. It's very different, and thinking about that. So that's super interesting field. Beautiful work's been done by so many.
So on the scheme of like how we imagine rodents in their social structures, there are mice and rats sort of comparable. I mean, I know it's much less of a.
I don't know that much about rats, but I think it's similar, but I actually don't know. I know for mice that they're similar people in a lot of ways, in that they're like kind of default group size 20 to 30, sort of like a default mouse colony in the wild. So not that different from like, classroom sports teams, departments kind of thing. And your rank is flexible, but pretty stable. Like once the rank and hierarchy is formed, it's like pretty stable. There's like different hierarchies that can be formed with different strains of mice and under different conditions that are ranging from like linear to more egalitarian or sometimes more despotic. I think, people also can like form different, like a variety of different structures. It's not always linear, it's not always one thing. It depends on the conditions and sort of the way that ranks are enforced and expressed, not dissimilar. So there's a lot of commonalities between people and mice. I think it's sort of interesting to compare to marmosets, where they really like kind of spend more time in the family unit, which is, another really, like, humans are so versatile, and across different phases of civilization, we've had more communal versus more nuclear family structured social groups. So I think studying these things across species is really important to do.
Yeah, no, that's what I was hoping that's what I was trying to get at there. But well, I think, should we call it, guys? This has been a lot of fun, and I could tell people are sort of sitting on their hands trying not to ask more questions. But we hope to hear more about this stuff. This is fantastic. It's been really timely. Really enjoyed getting up to speed on a topic I hadn't really read about at all until your work. And thank you for joining us, Kei Tai. This has been Neuroscientists Talk Shop.
Thank you so much for having me. It's been a pleasure.